Dear participant, welcome to the symposium and I will explain skeletal deformities of sparse aorta grown under various light intensities. Sparse aorta is one of the most important cultured species that are mostly found in Mediterranean country and also in our country. Uh, Seabream that are found in cultured conditions and often observed on skeletal abnormalities from hatcheries. The present study examined the bone and cartilage structure deformities of uh, sparse aorta that are used various light intensities within cylinder tanks. Four different experimental groups were constituted in this study. These groups are L3 2, L3 7, and C3 1 and C3 6. At the end of the experiment, 5,202 spermata were stained with allicin blue and alizarin red by using the double staining method in order to determine the deformants in the bone and cartilage structure. Finally, 758 deformed juvenile seabream were found among the uh, 5,202 juvenile seabream that were examined. The most deformation was observed in the juvenile fish in the C31 tank. Among the experimental groups, almost all of the deformities in the spine were found to tuberculosis in the Hamal region. You can see the study examined whether different light intensities have an effect on the scatter deformity in sparse aorta grown under various commercial farming conditions. The present study was conducted with the sparse aorta fry options from the Güvercinlik and Ören hatcheries in the Kılıç Marine Products Buffer Adaptation Unit. In the experiment, the effect of the light intensity on the skeletal deformity of the fishes when the seabream larvae with different tank volumes, different light intensities, and different growing origin arrived in the adaptation unit were tested in commercial environments. The experiment includes scientific and commercial deformity analysis. Morphology and scientific deformity analyses were conducted at the Aydın Adnan Menderes University, Faculty of Agriculture, Aquaculture Research and Training Units, while the commercial deformity analyses were conducted at the Kılıç Marine Products Buffer Adaptation Unit. Scientific deformity covered 12 different types of skeletal deformity in 5 body parts and determined the formation angles and location of the deformities in Spain. In the deformation analysis method used by companies in fish selling was analyzed and marked in the light tray indicated absence of deformities in the spine, head, gill, and dorsal region. All of the Sparsaurata were fed with some commercial power feed and in the scientific skeletal deformity analysis to determine the deformities in the bone cartilage structure, the double staining method was applied with the alizarin red and alcine blue. The difference among the groups were determined with the Tukey test following in the one-way variance analysis. SPSS 21 and Microsoft Excel were used in the static evaluation of the data. total 5,202 samples were tested to determine the deformity taking place in the Sparsaurata larva. In accordance with the results, 758 scatter deformities were determined with the general deformity rate of 14.6%. According to the analysis, the deformity rate of the larva from the L3-2 group was the lowest within the ranking of the group, while the C3-1 group was the group with the highest deformity rate. The lowest span deformity rate was observed in the C3-1 group, while the L3-7 group had the highest spin deformity rate. The lowest hit deformity rate was observed in the L3-2 group, while the C3-1 group had the highest hit deformity rate. Table 2. Dry whites of the samples taken in the beginning and the end of the experiment were calculated statistical, and the difference between groups were demonstrated. The statistic difference between the groups in the dried white figures on the samples taken in the beginning of the experiment was insignificant, while the L3-2 and L3-7 experiment groups were different than the other groups in the end of the experiment. At the end of the experiment, L3-2 and L3-7 experiment groups were different than the other groups. In the present study, lordosis was the most frequent type of dimorphity between the days after hatching of 64 and 100 in the hammer region among the fishes in the L3-2 group. This was followed by the heat deformity, cryptosis in the prehamal and hamal regions, and lordosis in the prehamal region. The L3-7 group has the highest deformity rate, and the most frequent type deformity in its anomalies was lordosis that was seen between 65 and 100 in the hamal region. This deformity was followed by the heat deformity, lordosis in the hamal region, and cryptosis in the prehamal region, and lordosis in the prehamal region. In C3-1, the third group, the frequent deformities were analyzed to be heat deformation, lordosis in hamal region, and cryptosis in prehamal region. In addition, lordosis was seen in the prehamal region in the 60th day. Cryptosis wasn't found in the hamal region between the days 63 and 88. In the larva in the C3-6 group, the most frequent deformity types are the lordosis and heat deformity seen in the hamal region. It was followed by the anomalies of cryptosis in the prehamal region and lordosis in the hamal and prehamal region. Figure 1. You can see, taking into consideration the rates of fish with spine deformity from the groups in the end of the 88 dah and 100 dah in the experiment, C3-1 was observed to have the highest rate of the fish with spine deformity and the highest rate of the total deformity with scientific and commercial deformity in all experiment groups. Upon comparing the detection method of commercial deformity and scientific deformity, both methods are similar in the detection deformity, while scientific deformity provides a more detailed but slower analysis possibility. No difference was found between the groups upon examining the angles of lordosis in the hamal region among the samples taken on the day of arrival to the buffer adaptation unit from two different hatcheries. In the end of the experiment, a difference was observed among the groups with respect to the lordosis angles, and the lowest lordosis angle was found in the C31 plank, figure 1. In all experiment groups, the lordosis angle in the beginning of the experiment is smaller than the dose found in the end of the experiment. This can be interpreted such that the deformity with a broader angle exciting in fishes became more evident in later days. The angle narrows down with age and becomes evident in lordosis, particularly in Hamel region. Among the experiment groups, almost all of the spine deformities were found to be lordosis in Hamel region. This lordosis was distributed between the 12th vertebrae and 18th vertebrae in all experiment groups, mainly between the 14th vertebra and 16th vertebra. In the analysis in the beginning and end of the experiment, an increase was observed in deformity rate and in the rate of the fishes with spine deformity in all experiment groups. Table 3. It was concluded that the light intensities used in the experiments were not effective in the formation of skeletal deformities during adaptation. And you can see figure 3, uh, lordosis scatter deformities with different angles observed in the spruce aorta in the end of the experiments. Thank you for listening.